Have you ever wondered where all the food that we consume goes? While some of it leaves our body, we absorb some nutrients from some, respire with some, and others we store within our bodies as fat or adipose tissues. These white adipose tissues are the general form of fats within our bodies. They are composed of a single large liquid body to store energy. They are also used to keep our bodies warm. That's why I'm always eating.
Eating, otherwise known as consumption of food, is a very important process to all living organisms. All of the food we consume mostly has nutrients that play very important roles in the normal functioning of the human body. For example, with a piece of pepperoni pizza, some of the main components are the pepperoni, which is the protein, which is broken down into peptides and amino acids, the cheese, which has the fat, which is broken down into lipids, and then further broken down into fatty acids and glycerol, and the crust, which is the carbs that is broken down into monomers such as glucose. All of those end parts are important in the body, especially with cell structure and other processes such as cell signaling, enzyme functioning, the transfer of hereditary information, and storage in the cell. Additionally, the food we eat has important vitamins and minerals, all of which play important roles in the cell membrane protein functions and in the prevention of other pathological diseases. The body is almost like an open system, since whatever is consumed is broken down and integrated. Additionally, if there's more input than output, consumption and required of food, the excess fats and carbohydrates will either be stored or converted to fat or excreted. If this process continues, this could potentially result in metabolic diseases such as obesity.
When I consume more energy than my body expends, I store that energy as fat. Only if I'm in a caloric deficit will I burn the fat away. It is hard for me to find the time to properly exercise and self-control to stop myself from eating all that ice cream. I could do surgery to remove that fat, but that's both expensive and unsustainable. I'll gain it all back. Unhealthy eating habits like bulimia would only further hurt me. If only there was a way to minimize the risk of obesity and other eating disorders.
On the contrary, obesity is a major problem, widely recognized as an epidemic that is affecting the whole world. It has been linked to causing cardiovascular diseases, strokes, type 2 diabetes, as well as certain types of cancers. In 2012, it is estimated that in the US alone, 34% of the adult population and up to 20% of the younger population are obese.
Breaking news! A new study by the American Journal of Physiology just shows that there's a way to minimize the risk of obesity. Researchers show that different effects of irisin in mice and how they can be minimized in the effects of obesity. The big question is that does irisin have the same effect in humans?
In fact, living mice turned white adipose cells, a form of fat cells that are burned, to brown once irisin was introduced to their systems and prevented them from becoming obese, even on high fatty foods. Another research done on humans revealed humans also have brown cells, but they disappeared very little as they get older, with exception to those physically active. In general, on a cellular level, brown cells showed no response to irisin, but white cells did.
Stay tuned to find out more about how the researchers specifically conducted their experiment.
Scientists acquired multiple fat tissue samples from female donors between the ages of 17 and 33. The collected fat tissues were cut into small 2 to 3 millimeter pieces and incubated in flasks to separate pre-adipocytes from mature adipocytes. The pre-adipocytes were incubated for 2 weeks to allow them to differentiate or grow into mature fat cells to be used for experiments. To demonstrate the effects of irisin on white fat tissues, the newly mature fat cells were separated into a 6-well plate and treated with a known concentration of irisin. After 4 days of incubation, the cells were stained red to better observe the white fat cells. When the scientists placed the tissues under the microscope for examination, they found that irisin-treated cells reduced in quantity. Fewer and smaller mature white fat cells were visible under the microscope compared to the untreated group. They found that irisin had a significant effect on the white fat cells, reducing the quantity by up to 60%. The scientists also found that irisin did cause browning effects on the white adipose tissues. They found that irisin increased protein levels related to brown fat cells, mainly mitochondrial uncoupling protein 1, or UCP1. UCP1 is the main cause for the increased heat production in brown fat cells by uncoupling or stopping the electron transport chain. While white fat cells contain a single lipid body, brown cells usually contain multiple lipid bodies and contain much more mitochondria, the main energy engine of cells, and coloring the cell brown. This is what gives brown cells their superior ability to generate heat, a major component of metabolism. The brown fat consumes more energy as heat, which would otherwise be stored as white fat cells in our body. Therefore, the scientists wanted to see how irisin could affect the metabolism rate of white fat cells. The mature adipocytes they grew were placed in microplates at various concentrations of irisin: no dose, which serves as the control group, low dose, medium dose, and high dose. The cell cultures were placed in a machine to determine oxygen consumption rate, also known as OCR. When the scientists observed the OCR, they found that irisin had a different effect depending on the concentration. In low dose, irisin didn't have much of an effect, but in medium and high doses, the basal metabolism increased. Basal metabolism is the energy your body uses to maintain itself. It is something going on right now as you rest and watch this video. This would indicate that irisin-treated cells would burn more energy than non-treated white fat cells. In addition, high-dose irisin was also found to increase maximal metabolism, or the maximum energy released by the cells. All of these factors combined would in turn help bring us closer to a caloric deficit
and ultimately weight loss.
While further experimentation is required before fully understanding the effectiveness of irisin in reducing human obesity, the study has shown the great promise and new potential for treatment options for obesity. Conflicting research has shown that irisin's effectiveness on turning white fat cells into brown fat cells depends on the cell's maturity, failing to work on non-mature cells. In addition, more research will be needed before clinical trials to better understand the lasting effects of irisin.